Okay, now Linda is going to speak.
I just want to say welcome to everybody, and we're sorry that we had some difficulties here, some technical difficulties, but we're ready to go now. And this is going to be the first of hopefully a number of events that we're having in collaboration with the San Francisco AIDS Foundation. We are from the Martin Delaney Collaboratory, from DARE, from RID, from HOPE. Hope is especially going to be featured tonight with Melanie Ott. But we just are very grateful that you've come. Happy birthday to the March guys. And that's pretty much all I want to say. What we're going to do is have Melanie give her presentation. We're going to ask her a couple of questions. We're going to open it up generally to, the group to ask their questions. And it's going to be hopefully pretty simple, as simple as that. We're going to do an evaluation about midway, three-quarters of the way through that hopefully you can just tell us whether you liked it or you didn't. And that's it for us, really. Again, thank you for coming. And that's all I want to say. Patricia, you want to introduce Melanie? That would be very helpful.
My name is Patricia de Fischere. Welcome, everybody. I'm so happy to be here at SFAF again, and I have the immense privilege and honor to introduce Dr. Can you hear? You cannot hear me still, even with the microphone?
We
All
can hear
Can hear you.
at the back. Okay. So it's a huge honor to have Dr. Melanie Ott with us here today. Melanie is the director of the Gladstone Infectious Disease Institute here in San Francisco. She's also the vice president of the Gladstone Institute. For folks who don't know, the Gladstone Institute is a research institute here in Mission Bay that's devoted to addressing many diseases area, neurology, cardiology, and also virology and immunology. And Melanie is also the program director of the HOPE Martin Delaney Collaboratory. So for folks who are not familiar, the Martin Delaney Collaboratory are an effort from the NIH, National Institute of Health, to find and develop cures for HIV. And the HOPE Collaboratory is a kind of a new kid on the block, if you wish, who's going to develop a novel approach to cure HIV using a technique which is called silencing. And you're all going to learn tonight what silencing is about. So I will let Melanie, maybe highlight some of the things in her career which brought her to where she is today. But Melanie is not only an incredible scientist who's devoted her life. She's a medical doctor trained in neurology. She's an incredible human being who's very dedicated to community education, engagement, and listening to community voices. And she is recently a grandmother to Esme and Niall. And without further ado, I bring to you Melanie Ott. It's really a privilege to have her speak to us today about how to cure HIV using silencing approach. Welcome, Melanie.
Yes, Melanie, I think it might be helpful when you start to speak if you don't move.
Yes. Okay. So let me see. Do you hear me?
Yes.
You can hear me if I leave it like this?
Yes.
Oh, but not you guys. You guys hear me now? No?
We can hear you on the Zoom.
Okay. Can you hear me in the room? Okay. So let's try this. Okay. I'm not moving. I'm going to be speaking into my multiple microphones here, and somehow we will manage. Okay. Anyway, I'm really excited to be here to meet you all. I'm very excited to meet all the people on the Zoom. Patricia, thank you so much for your kind introduction. And yes, I'm a scientist. I have been working on HIV for over 30 years, and I have been really devoted to finding ways how to turn the virus on and off. And I think that's what I want to talk to you today. And so you will hear about why we need a cure, why silencing is an interesting new approach, and how can we move this forward, and how does that connect to what we do otherwise in the science field today. I will give you a little snippet of how our
Melanie's going in and out.
connects with research and what we can do to get younger and potentially take care of the virus. So this is
Melanie's volume keeps going in and out.
sneak this.
Cannot understand.
Understand
Can't
now?
hear you, Melanie. You keep going in and out.
You hear me now?
Yes.
Yes.
Okay, so I keep this on here. Okay, let's start. So let's go for the first slide. Not yet there. Anyway, my first slide should tell you that HIV is not new to our body. HIV is a so-called retrovirus. I don't know if you've heard about this. It's a virus that can come into the body and then integrate into our genome, just as if.
Oh, there you go. We got it now.
HIV is not new. We have 60,000 viruses like HIV.
The volume is not good.
Oh, God.
Yeah, it's kind of
Going
bad.
in and out.
Really bad. We can't understand a word.
Well, I cannot do anything else.
So that's good. Right there is good. Whatever you're doing right there now is good.
Okay, I have to ignore you guys on this side, okay? It's not by purpose. I will stay like this so that people on Zoom can hear. Anyway, HIV is not new. HIV is one of 60,000 ancient retroviruses that we have in our genome that came in our genome millions of years ago. Millions of years ago. So we are very familiar with this, and our body is very familiar because it has actually developed very precise mechanisms how to deal with these 60,000 retroviruses in our genome. It has actually our body has evolved to devote a whole series of specific mechanisms to silence and deal with these ancient retroviruses and keep them silent, keep them quiet. They're still in our body. They're part of our genome. They're actually about 10% of our genome is coming from old viruses. And these are viruses like HIV. And we know how we deal with these ancient retroviruses. And we think that we can learn from this how we should deal with HIV. Now, we don't want to wait millions of years to evolve the mechanism to silence this virus like we have done with other viruses. We want to do this faster. And we want to use the same machinery that our body has basically devoted to it and the same principle, but do it medically, do it faster, accelerate it so that we can put this virus into the same state that all the other viruses are in, that we already contain, that everybody on the world contains. So this is nothing special about certain people. It is something that has evolved to be organically part of us. And this is basically what we're trying to do with HIV, which is a new retrovirus that our body has not yet learned how to deal with. But in principle, we in the HOPE Collaboratory want to do exactly the same thing. And the problem that the point is that HIV is not yet dealt with like with all the other retroviruses we see because HIV is not silent. We need antiretroviral therapy to suppress the virus. But when we stop the antiretroviral therapy, the virus comes back because it is not silent. Endogenous retroviruses or others, they basically stay silent, whatever you do to them, and they don't need specific treatment. And that's exactly what we're trying to achieve. If I can advance. Nope. No?
Nope.
One second.
There you go.
Okay. And the reason, so that the reason or the loophole that we want to fill is basically that antiretroviral therapy is wonderful. We all know this, and nobody wants to criticize it. It addresses a lot of stages in the virus life cycle, but it also has some loopholes. where it does not have drugs that are, specifically devoted to it. We all know it's a cocktail, different drugs that are working together. But one part is not yet addressed in this drug regimen, and that is called transcription. That's a scientific term, but what it means is just the engine that basically produces new copies of the virus in the cell. That is called transcription. That is still ongoing. Despite antiretroviral therapy, it cannot form any viruses because all the other steps are blocked. But the pure fact that these genomes are being produced is an activity sign that is still there, and it has specific consequences that I want to discuss with you today. And so what we try to do in HOPE is basically close this loophole and find new drugs that we can combine with the antiretroviral therapy so that that step in the viral life cycle is achieved. And that is leading to that silencing step that I've told you at the beginning, which we have seen with other with natural endogenous retroviruses in our body. And that's our basically goalpost where we want to go. And that we think that if we add specific things to antiretroviral therapy, how we have it today, we can potentially cure it. And curative in the way that when we stop it, the virus is silenced, so it does not come back, and we can, continue, and the virus is controlled. I just have a few slides for you, give you a little bit of the background why we need a cure today. You all know this, so I don't have to, ponder of it. But I want to point out again that in the emergence of antiretroviral therapy 40 years ago was really an absolute milestone in the lives of everybody, but also specifically for me as a virologist, because it has really inspired so much important work, not only on HIV, but also on other viruses. Because before these antiretroviral therapies, the idea was that viruses are only treated by vaccines. So if you want to treat a virus or you want to prevent a virus, you have a vaccine. If you want to if you treat a bacterial infection, you all had strep throats or whatever, you get antibiotics. So treatment is reserved for bacteria, but not for viruses. And this has been completely flipped upside down because we now make antivirals for many, many viruses, and HIV was the trailblazer here with the enormous success of antiretroviral therapy. I want to give a shout out to Gladstone and to my friend Patricia. I don't know where she went. There, she's eating cake. Because, of course, antiretroviral therapy not only has revolutionized the treatment, it has also revolutionized prevention, because we know that a vaccine for HIV incredibly difficult to achieve because the virus is so variable. It can really sneak around every immunological, obstacle that we put in its way. But we have also learned with this landmark publication and study that was led by Patricia and Bob Grant at the Gladstone Institute in 2010 that PrEP, exposure prophylaxis, is possible. And of course, this landmark paper has led to the success that we have today with PrEP, where we have lenacapavir just being launched by Gilead, and that you can get 2 shots a year and have protection. So that's almost a vaccine if you want to talk about like a yearly flu shot, So this is an enormous progress and really good use of what we have currently with antiretroviral therapy. But we also know that we are facing a lot of headwinds, especially today. And I say here, a cure for HIV has never been more urgent. Lifelong antiretroviral therapy is no longer guaranteed. We know that we cut the support for Africa last year with PEPFAR, and we know that there's a lot of cuts in the States for support for HIV meds. And I think this is something that I think if we can now accelerate a cure where intake of drugs of ART doesn't have to be daily or long term. I think it is a good incentive to do this now. I want to remind you that we already had cured people. We have people that have gotten rid of the virus, and this is Timothy Brown, the so-called Berlin patient who lived a long time here in San Francisco and was treated here at SFGH, who was cured, the first person who was cured by a bone marrow transplantation, which is a very invasive procedure. And actually, Timothy had to do it twice and almost died every time he had to do it because he had rejections. But he was transplanted with the T cells that were immune, with an immune system that cannot be infected by HIV, and that basically cured him. And now we are up to 11 people who have been cured with this. Now, this is a big number, but it's also a very small number. And this article in the New York Times, after the second cured person, actually argued that it was not the cure for my HIV because or not for that person's HIV, because it's not possible to do this for everybody, and it is a very invasive procedure. And you have to be lucky to have a match between a donor that has actually that resistant immune system that would not be reinfected with HIV. But it has taught us a lot, but it is a very unpractical approach, let's say this. I think I can skip this. I just wanted to say that what I told you about transcription, that it is still active despite a current antiretroviral therapy, we think is an important point, because it is not only, and that's what I skipped here in the slides, it's not only determining how fast a rebound happens if antiretroviral therapy is stopped. And it's very easy to understand. If the virus is simmering and has activity, and you take away the break, which is the treatment, it can come back much faster than if you have it very deeply suppressed and there's very little simmering of the virus and activity. Then it takes longer until the virus reawakes. But what this transcriptional activity, or that simmering of the virus, is doing is also that it stimulates the immune system. Our immune system is very, very sensitive. It's actually one of the most sensitive detection methods that we have in the body, because it really finds things very, very, very diligently and very, very sensitively. So what happens is that even if these genomes are being replicated and not producing any virus, the body senses that something is wrong. And this is not only with HIV, we now know also with other diseases. We think that long COVID, for example, it's an immunological disease that is caused by potentially a few copies of the genome hanging around in the body, in basically igniting the immune system to be continuously active. So this leads to sort of inflammation, and we all have learned that inflammation is not good. We want to keep all our inflammation levels down. And so with this, with this intervention that we're planning, we also planning to basically suppress basically that simmering of the virus at its root causes. And I made this like a little bit of a tree here, saying that the genome is the one that is the root cause of all of it, because it gets replicated, gets sensed by the immune system. It can produce some proteins that can be also detected, and we have different interventions that we do against the individual ones. But we were proposing that we really have to go after the root cause. We shut down the simmering, because then you have no worries about, being your immune system being activated and potentially, causing some bad things on the long run here. And again, that's what we want to do. I want to point out, I think you brought some copies, Patricia. I'm enormously grateful for this wonderful book, Holding HIV, that our community group has produced. And they have these were study participants that were specifically talking about the silencing approach, because one of the flip sides of the silencing approach is that the virus is still in you. It's not going to be eliminated. It is basically still there, but it's silent. And as I said, we have 10% of our genome in the same state, an old virus that came in and that we silent naturally. And I think that's what this poetry book is sort of dealing with, and it has absolutely wonderful gems in it, and I read in it quite often, because it really sort of shows the emotions, the feelings, and how to deal with this idea of HIV being dealt with in that silencing manner. Now, last in my talk, I want to talk about what is actually the silencing and how do we want to do this. I want to give you a little bit, and this might be a complicated slide, but my husband is an aging researcher, very much interested in rejuvenation and longevity and all this. And one of the things that the aging field currently is doing, it's trying to measure our age. We all measure our age with our numbers of years, but we also know that people age differently and that there's different factors that are playing a role how we age. There's actually just the fact of women versus men age differently. And one thing how we're trying to capture this, now the aging people, they also need this, because when they want to study interventions, they cannot wait 60 years to see if somebody is living longer or not. They have to find sort of a biomarker, whether they reverse or accelerate this aging process that they're studying. And it is actually a very interesting principle that they have come to, which is the DNA. We all have our genome, our DNA. We know that HIV is in there, but the DNA is not naked. The DNA is actually decorated with all little tiny things that get turned on and off onto this. It's called the epigenome, the genome that is around. And one of the most important things are these little balloons or lollipops that I put up here, which are so-called methylation marks. So what happens is that genomes accumulate these little decorations differently during the lifespan. And we know that during aging, there's a change in this, which is sort of tried to be represented here. Some of them are being removed and some of them are becoming more active. Now, but so this is what they call a clock in the aging field. You can make this methylation clock, and everybody can basically test this now. They test certain methylation clocks or certain methylation marks on the genome, and they find that and then they can basically test potential slowing factors like physical activity, healthy diet, good sleep, many friends like you have here. That is all slowing factors. But of course, accelerating factors are smoking, alcohol, unfortunately, stress, infections, and other things. So this is what the aging people are testing with their clocks. Now, we are very interested in that because the methylation marks is really the way how we silence our ancient retroviruses. It's really this whole concept of methylating your DNA or putting these little lollipops on your DNA is coming from our genome, having evolved to actually shut down certain areas in our genome, including these ancient retroviruses. So this is exactly, ancient retroviruses are silenced by methylation, by these lollipops. So we basically want to put these methylation marks specifically on HIV to silence it down, because HIV is not yet silenced, as I told you, because it evades this methylation and can rebound because it's not basically shut down like the other and retroviruses in our body. And the interesting part is that if we really look back into these ancient retroviruses again, we have learned everything we know about methylation and what the aging people have now basically adopted from these old retroviruses. They have these specific factors that are called, they're looking like a crab here because they're called crab zinc fingers. And these crabs, we have about 400 of them in our body that we have specifically developed only to silence these ancient retroviruses. They are specific armada of proteins that look similar because they all recruit these factors here that I pointed out here, but they basically sit on these endogenous retroviruses, put the methylation marks, and keep it silent. That's what it is. But we have not yet a crab zinc finger for HIV because it's not long enough in our body that our body has basically had a time to evolve something slow like building a new protein that is specifically devoted. So what we want to do is we want to build interventions that basically mimic these crab zinc fingers that are specific for HIV so that we can basically do exactly the same thing that we do with endogenous retroviruses, but just in a medical fashion. So we don't have to wait a million years until it happens by itself. And this is just to show you how it actually looked in our research. These are all methylation marks. This is the HIV genome that is integrated in the body or in the genome. And this is actually the marks that we basically force because we put a specific intervention into it. And this is what we're looking for because that is what keeps the virus basically down and prevents it from coming back. Now, this again, we're doing this for HIV, but it is also caught or went viral. I said silencing goes viral because it has also been now applied for other chronic viruses. And we know they have HIV as a chronic virus, but we have, for example, papilloma viruses that we have in our body. We have hepatitis B, which is in livers. Very, very, prominent chronic virus that causes a lot of havoc and causes cancer in many people. We have herpes viruses that stay with us all our life. All these viruses have the same principle. They simmer, also they keep silent so that they're not being eliminated, but they simmer. And that simmering is causing problems. In the case of hepatitis B, it causes cancer in the liver. In the case of herpes, when it comes back, it causes blisters or it causes fatigue in people. So there's a lot of these same principles with other viruses that we carry. And so, but these are different viruses. They're not retroviruses like HIV, but still people applying the same principle. They're using the machinery for endogenous retroviruses devoted to these chronic viruses and try to silence it. And that's what this clinical trial in Hong Kong for hepatitis B, where they developed an epigenetic silencer to do this. Now, an epigenetic silencer is basically a gene therapy thing. It's an enzyme which basically is similar to the complex that I showed you. It uses the CRISPR technology, which is a Nobel Prize winning technology, which helps us to put something very precisely into the genome. And so people are using this because you can very precisely target it to HIV, or you can target it to hepatitis B, and you can basically put these little marks on the genome, and then the genome is not active anymore. This works very well for the liver because, and this is done with one injection. So you don't need to do this many times because once you bring the machinery in, it does its magic, it puts the methylation marks, and then it disappears. You might have to refresh it, but this is basically the principle. You do it once, and it leaves a lasting effect, basically. Now, when you do this and you get an injection and whatever, everything ends up in the liver. So everybody, all these companies, they're focusing on the liver because the liver is a wonderful target for this, because You don't have to be very inventive. You just put it in your body, and the liver will eventually absorb a lot of it. And it's exactly where the people want it to be, because that's where the virus is. Now, HIV is a little more complicated because we know it's not a liver virus. We know that we have these like residual, very rare reservoir cells. That's basically the cells that still contain replication competent virus, but that is basically everywhere in the body. So it's much more complicated for us to target whatever we put in there exactly to the cells that we want, because it's not necessarily in the liver. And so we pursue two approaches. We're pursuing the gene therapy approach, like I told you, which is very straightforward, and we can do this on cells, mice, and many things very, very efficiently. But we still have to find a very sophisticated way to reach each of these cells everywhere in the body, basically. So that's the challenge here. What we think is much easier is to build a drug. So we can really put it to the antiretroviral therapy cocktail. Much easier, no injection, no nothing, you know, and it will do the same thing. But the bar is a little higher for this, because you have to make sure that that drug is specific for HIV and is not going and silencing everything left and right in your body, because that's what you need still. And so we have a whole pipeline that we have basically built, where we're testing known drugs that we have found that are clinically approved that do silencing of HIV on top of what else they're doing. We have these nucleic acid, these gene therapy approaches that we have done that are very specific. And we're doing a very interesting and super exciting approach currently with world-class pharmacologists and chemists, where we basically take known silencing drugs and couple them with something that would specifically bring them to the HIV promoter. So that's sort of the three approaches that we're using. But I want to tell you, sorry, I'm pushing here something wrong. Can this go away? Okay, thank you. But I want to specifically focus on one thing, which is, oh no, doesn't shut down, which is something from my colleague Nadia Rowan, who is at the Gladstone Institute. She just a week ago published this paper in Immunity, and I'm circled here the word metformin. Who of you have heard the word metformin? Yes, very good, very good. So it's a very interesting drug. But what Nadia did here, she used sort of samples from people who underwent treatment interruption. And she got the samples before and after they did this. And then she basically did a whole bunch of analysis to find out what determines if somebody rebounds early or later. And she found two factors. One is the zinc finger, which is exactly that crab thing that I told you about, that is exactly sitting on these endogenous retroviruses. And the other one is another factor, complicated, but it's the target of metformin in the cell. So metformin basically manipulates this factor, and we know that this factor is basically determining how fast somebody rebounds or not. And so metformin is, of course, a drug that is very well known. It's an ancient drug. 1920, people have started to think about compounds from metformin. It's coming from this French Lilly, where it was identified. They actually used teas from this, I think, to diabetes-like symptoms. And of course, metformin has been approved in the United States as a type 2 diabetes drug. And you see these are very similar structures to this plant thing. And I think this person here, Jean Stern in France, was the first one in the 50s to find out that metformin would actually be beneficial to suppress diabetes symptoms. It was approved in France for treating diabetes in 57, and it took another 40 years until it was approved in America. But it is one of the most widely spread drugs that people are taking. Now, the biggest jump this drug has experienced is in the longevity field, because now people have found out that if you give metformin to mice and also to people and to worms, they live longer. And I don't think they have done this in people yet. There's a big trial that they're still waiting to be, but they have shown this in monkeys. And another interesting thing is that metformin is actually a very good drug that prevents long COVID. So a very interesting drug with a lot of unexpected side effects, positive side effects, basically. But I would say there's a lot of people, because it's dead cheap, it is out of patent, and it's very easily and very well tolerated, that a lot of people are on metformin, not only for diabetes and improving their metabolic health, but also for potentially improving their lifespan. Now, the interesting thing is that metformin decelerates the aging clock. So it changes your methylation pattern, which is something that we like, because remember, we want to manipulate methylation to silence HIV. And so we think that metformin might not be a dead ceiling silencer, but it's definitively a drug that works in the pathway that we like to achieve, which is turning down the simmering of the virus despite antiretroviral therapy, putting these methylation marks onto the DNA and shutting it down. And that's exactly what we're currently testing in mouse models. And so this is an example of a drug that we are using, which is freely available, to basically achieve this goal of closing the loophole here and finding new drugs that actually silence transcription or silencing the replication of genomes. And that would be an HIV specific inhibitor. And the point is, again, as I said, if we shut this down and do this with mechanisms like the methylation clock that I've shown you, these are permanent or very long-lived marks that we don't have to continuously refresh by taking in the drug or the gene therapy. We can do this once and we have the marks, so that we think that eventually this whole tree of simmering that I had described to you is going to wither and die, and we don't have to worry about it anymore. So there's the sign in New York that I loved. So I think there's some hope that we think is alive. And I don't want to end my talk without acknowledging our wonderful community engagement group that we have at HOPE and at Gladstone, a really fantastic group that keep, all of you informed and include and engage a lot of our young scientists. We have podcasts, we have visits in the lab, we have dancing together, we have a lot of wonderful interactions. They have produced, they might get an Emmy. They have submitted a really interesting short movie on Jim from San Diego, who devoted his body to research in the last gift cohort, and actually his last gift is living on in the Gladstone with us. And I want to thank the whole HOPE community, really incredibly devoted people, mixed with a lot of our community engagement people. And I don't know if this is going to work, but this was a movie of the Gladstone Institute 5th floor where we reside. Maybe not possible. It was a beautiful drone that was flying across our BSL-3 facility, which is the biosafety facility, and up into the sky of San Francisco, but maybe next week. And with this, I stop and thank you all for your attention.